Good morning, students. Now in the today's period, we can study here that how the protein is produced. You know that protein is an important biomolecule and it is a need to produce the proteins again and again because it requires for the new cell. Here in protein synthesis, two processes are important. One is called as transcription process and another is called as the translation process. What is transcription? We know that there is a DNA molecule and DNA has two templates. So the template, when it is able to produce the strand of mRNA among the both strands, one strand is acting as template and it gives its coded genetic information on the mRNA. So it is called as the transcription process. That is, the copying the genetic information from one DNA strand to single DNA or single RNA or single stranded RNA, that is the transcription process. Again, keep in your mind that production of protein is must because cell needs protein again and again for the different purposes, for hormone formation, for the growth, for preparation of DNA, etc. And here two steps are important in protein synthesis. These are transcription and translation. Transcription means the copying genetic information from one DNA strand to single stranded RNA. So the copy is made on one strand. That strand is called as template and genetic information is transferred to mRNA. Now what is the central dogma of molecular biology? In the molecular biology, DNA is producing the mRNA. The process is called as transcription process. And from the mRNA, the language is in coded form. The synthesis of polypeptide chain takes place or proteins takes place, that is called as translation. We know the translation. Coded language is translated into the formation of polypeptide chain and that is the central dogma of the protein synthesis. And here the mRNA, that is from DNA to mRNA, the transcription process takes place. mRNA is produced, that is working as the messenger for the protein synthesis. And this type of transfer of unidirectional information from the DNA to mRNA to protein synthesis is called as the central dogma of molecular biology. What is central dogma of molecular biology? That is the messages which are given by the DNA on the RNA and formation of polypeptide chain takes place. That is the central dogma of the molecular biology. This type of work is given in the or studied in the retrovirus in 1970 by Temin and Baltimore. These are two scientists, Temin and Baltimore. They had given their study or studied in the retrovirus in 1970. Now we can study here the different processes in detailed manner. We know that number one process called as transcription, that from one DNA strand the information is copied and then it is transferred in mRNA. Here enzyme RNA polymerase is useful. We are studying here transcription process on one DNA strand. It works as a template. The genetic information is copied and transferred in mRNA with the help of RNA polymerase enzyme. Now the DNA is located inside the nucleus. So here transcription takes place in the nucleus, and translation means formation of proteins takes place inside the cytoplasm, and then the information is transferred toward the ribosomes, which are present in the cytoplasm in endoplasmic reticulum. These are the smaller cell organelles which are present inside the cytoplasm. See here, nucleus consisting of the DNA transcription, mRNA formation takes place, and it is transferred to cytoplasm, and the formation of protein takes place on the ribosome through the transferred genetical language on mRNA. So ribosomes are also important in the protein synthesis. Now it occurs in the nucleus in Z1 and Z2 phases of cell cycle, where it occurs in the Z1 gap 1 and gap 2 phases of the cell cycle. So here we can study here that it has two sides, DNA promoter and DNA terminator side. Promoter to promote or to produce, terminator that is the end शेवट आहे त्या प्रोसेसचा. सो इट हॅज promoter and terminator sides. So initiation, there are three steps: initiation, elongation and termination. So these are the three steps involved in the protein synthesis: initiation, elongation and the termination. So see here, this is the transcription rate. What the transcription unit has? It has two segment of DNA having 5' end, 3' end. Second having another side that is 5' end and 3' end. This is the promoter side. This is the promoter side. This is the coding strand. Here we get the structural gene, and now here the template is working there. That is, the template strand is working there, and here termination takes place. Now each transcribed segment of DNA, is the transcription unit. What is the transcription unit? Each transcribed segment of DNA is transcription unit. It has promoter, structural gene and terminator. Now the two strands of DNA in structural gene shows promoter at 5' end. Promoter at the 5' end, and here it provides the binding site for RNA polymerase enzyme. So binding site where the proteins are produced. So it is the binding site for the enzyme. Now here the 3' end having the terminator site. Now the structural genes are present here. It has the structural gene has we find that it has two strands of DNA has opposite polarity. They have opposite polarity. So here the DNA strand having 3' to 5', 3' to 5' polarity, it acts as a template. 3' to 5' polarity, it acts as a template. What is template? It is a strand, एक स्ट्रँड आहे डीएनएचा, जो की त्याची जेनेटिक इन्फॉर्मेशनची कॉपी ही mRNAच्यावर काय होते, ती ट्रान्सफर होते. दॅट स्ट्रँड इज कॉल्ड ॲज द टेम्पलेट. सो हियर इन द ट्रान्सक्रिप्शन unit, here promoter site is there, structural genes are there, and the termination end is also there. Terminator is located at 3' end, and so it is called a downstream also. It defines the end of transcription. Key process. What is transcription? That is the transfer of genetic information from DNA to mRNA. So this terminator site or the terminator, it decides the process or end of the transcription process. After binding of promoter, RNA polymerase moves along the DNA. After the binding of promoter, RNA polymerase moves along the DNA, unwinding takes place and one strand acts as template and other as coding strand. One is acting as template and other is coding strand. Ribosome triphosphates joins to the base of DNA. Ribosome triphosphates joins to the base of DNA template chain, and as the transcription process, the hybrid DNA RNA molecule dissociate. The RNA molecule dissociate. Dissociate means become separate and mRNA becomes free. When RNA goes to transcribe region of DNA, molecule becomes spirally coiled and attain the double helix structure. So we will find that here in eukaryote, 3 types of polymerase enzymes are found. 3 types of polymerase enzymes are found. RNA polymerase first, it transcribes rRNA, ribosomal RNA. So see here, what are their functions? In the eukaryote, 3 types of RNA polymerase are found. Number 1 is RNA polymerase first, it transcribes rRNA. Now the second is RNA polymerase second, it transcribes mRNA and heterogeneous nuclear RNA. Heterogeneous nuclear RNA, that is hnRNA. So RNA polymerase second transcribes mRNA and hnRNA. Now the third types of enzyme, RNA polymerase third, it transcribes tRNA, that is the transfer RNA, and smaller nuclear RNA, that is called as snRNA. So in short, we have studied or taken information about the protein synthesis, that it having transcription and translation process. Transcription takes place on the DNA strand, that one strand acts as template, mRNA is produced, the genetic information is transferred on it and it reaches to the ribosomes, and there the protein synthesis takes place. Here the 3 types of polymerase enzymes are useful. RNA polymerase enzyme first, useful for formation of rRNA. RNA polymerase enzyme second, useful for formation of mRNA and hnRNA. RNA polymerase enzyme third, it transfers tRNA, and snRNA, that is the smaller nuclear RNA. Thank you.